Okay, good morning, ladies and gentlemen. We can start. It's my great pleasure and honor to introduce and welcome our speaker today, Professor Stefano Cappa. Stefano was awarded a degree in medicine and a specialization in neurology at the University of Milan. And he has held clinical and research at the Neurological Clinic at the University of Milan and the Division of Neurology at the Niguarda Hospital. And he spent periods of study and research at various research institutions such as University of Boston, MIT, the MRC Cyclotron Unit Hammersmith Hospital in London, and the Max Planck Institute for Cognitive Neuroscience in Leipzig. Since November 1999, he has been professor of neuropsychology at the University of San Raffaele in Milan, and he also the director of the Division of Neurology at San Raffaele Hospital. He is now at the Pavia University Research Institute, USS. The research activities of Professor Cappa include the study of language disorders acquired after brain injury as a source of information and the organization of language and its related neurobiological disorders of memory in normal aging and in Alzheimer's disease. And finally, disorders of space resulting from brain injury. In particular, he is an authority on the study of unilateral spatial neglect. So we are very happy to have Stefano here today. The title of the talk is Cognitive Neuroscience and Dementia Phenotyping. Please, Stefano.
It's a pleasure to be here. I'm going to talk about some recent applied research we've been doing in Milan. What do I mean by applied research? I've been working, as Giorgio has been kindly summarizing, in several areas of cognitive neuroscience, keeping the clinical side that's sort of separated from my basic research for many years. Then in the last few years, I think that there is now some interesting comeback of the clinical applications of cognitive neuropsychology. And this is mostly in the field of neurodegenerative disorders. If you have a look at this slide, you have three classic areas of cognitive neuroscience research. That is the study of episodic memory, the study of language, and the study of social cognition. You all know that many advances have taken place in our understanding of these functions and of their brain organization. If you think about it, these three areas map very nicely to three areas of neurological research, which are now considered to be a sort of priority for many reasons. Reason number one, of course, as you all know, is the aging of the population. I'm not going to tell you the usual stories about the fact that the aging of the population has a huge impact on the prevalence of neurodegenerative disorders. Look, for example, at these three areas. The study of episodic memory clearly has implications for Alzheimer's disease. That is the most common dementia form. The study of language has important implication for another area, which is the group of disorders which are called primary progressive aphasia. That is the neurodegenerative forms of language disorders, which are often due to another condition, pathological condition, which is the frontotemporal dementia spectrum, but can also be due to Alzheimer's disease. In this case, we talk about atypical Alzheimer's disease. And the third field, which is probably the most exciting for some of us because it's sort of new and we sort of like new things. It's the field of social cognition, which has impact for still another form of neurodegenerative disorder, which is the behavioral variant of frontotemporal dementia. Again, this is typically due to a different pathology, but can also be due to Alzheimer's disease, atypical Alzheimer's disease. So I have examples for So I have examples, much better. I have examples for the three areas. I don't know whether I have time to go into all of this. I prepare more stuff as usual, as well, what probably I can talk about. But let's see how it goes. How much time do I have? 45 minutes, okay. So let's start with the episodic memory. If you show this slide to a group of people who are interested in Alzheimer's disease, they usually say, come on, because it's shown at each and every conference. But, so it's really boring. But what I wanted to point out is that this is the supposed timeline of the different events leading to clinically overt Alzheimer's disease. And what I would like to point out is that cognition is considered to be a late change, much later than the other biomarkers. The other biomarkers, for those of you who are not expert in the field, are typically CSF changes and neuroimaging. In particular, magnetic resonance imaging and PET. And in the last couple of years, a lot of interest has been devoted to amyloid PET imaging. I'm not going to talk about this, but I just wanted to point out that this late change in cognition, it's quite a clinical problem. This means that if you want to do an early diagnosis, what can you do? You must study asymptomatic people, do screening. Of course, there are many good reasons not to do this. It's a very common disorder. So what you would really like to do is to have sensitive and specific neuropsychological markers in order to decide who's going to get all the biomarkers. Otherwise, you run into big risks of overspending, false positive diagnosis, etc. So it's sort of paradoxical, but the high tech of the last few years has been resurrecting the interest in very low-tech things, such as neuropsychological tests. And this is quite new because what we know is that there are some very early events. This is another study which is always cited by one of the gurus of neuropathology, Heiko Brack, in which he was able to study a large number of brains of people who were not affected by Alzheimer's disease, and was able to find some very early changes affecting the hippocampus already in people under 30, in a minority of people under 30. And of course, this is an interpretation. This might indicate that a very early pathological process is starting in people who might later develop Alzheimer's disease. Why I'm showing you this? Because the ratio is that you should really try to find neuropsychological markers which are sensitive to dysfunction of this very focal damage to the hippocampal region, especially to this region, which is the transentorhinal cortex. And here is where cognitive neuroscience gets in. Because of course, we are aware that, in particular in the field of animal study, experimental neuropsychology, but also in the study of human subjects, there has been a big effort to try to characterize the consequence of disorders affecting this part of the brain. And this was not reflected in the traditional neuropsychology of Alzheimer's disease. These are the old diagnostic criteria. And you might see that basically everything goes. There is a list of tests of memory, and the idea is that any test of memory is good enough to make a diagnosis of Alzheimer's disease. This is probably true for clinically overt Alzheimer's disease, but it's probably not true for very early stages and for localized selective damage to the hippocampal region. If you look at the more recent criteria, the American criteria, more recent, but still, you see that there is a long list of memory tests, which are considered to be possibly adequate for the assessment of memory for patients in the early stages. There is a difference with the so-called European criteria, in which there is the idea that the impairment of memory should be found in specific tests which are supposed to be sensitive to hippocampal dysfunction. The claim has been made by a working group, and what they mean by specific memory test is a test which controls for encoding a recall, something which is not present in many of the traditional tests. The test is well, this is the version we standardized in Italian. This is controlling for encoding just means this. In the encoding phase, you ask a question, the patient has to point a name to the item. And this way you are sure that the item is encoded. Controlling for retrieval just means that in the recall phase, if the subject does not volunteer the name of one of the items, you give a cue. It was an animal. And this controls for retrieval failures. It's very, very, very simple. The basic idea is that this should make the test not more sensitive. Actually, it makes it less sensitive because it's easier, but it makes it more specific for Alzheimer's disease. The problem is that the claim is just based on a couple of studies and needs to be replicated because there are also other possible candidates. Another interesting candidate has been proposed by Sergio Della Sala, and it's actually not a long-term memory test. It's a short-term memory test. That is something which is usually considered not to be affected in early Alzheimer's disease, but it deals with visual memory. And he published some very promising data based on a very important population from the point of view of specificity. That is, patients with genetic Alzheimer's disease, carriers of the mutation, who are going to develop. It's a percentage mutation with 100% penetrance. So these are people who are going to develop Alzheimer's disease. Again, this is just a study on a specific population. What we are doing now, together with several people, is try to put these claims to test. That is, studying a large population and comparing the test in terms of sensitivity, specificity, and predictive value, which is the most important point. That is, how good is the test to predict the development of Alzheimer's disease? Unfortunately, I don't have the complete results. I can just show you that the study is going on, but I just wanted to share with you some very preliminary result. This is the comparison between the test which controls for encoding and retrieval, that is the free and Q selective reminding test, and this is the De La Sala test. And the bars represent the likelihood ratio, that is how useful is how predictive is the value, the value of the test for getting the disease, and this is the positive predictive value, or for excluding the disease on the basis of follow-up. And these are already quite interesting because, as you can see, these are the 3 of the scores you get from the La Sala test, the binding test, and this is the one of the scores you get from the free and Q selective reminding test. And as you can see, they have quite good positive likelihood ratio. And this is particularly good for this test, which is quite unexpected because this is a visual short-term memory test. It's actually better than the free and Q selective reminding test. And all three, but in particular the binding, are excellent from the point of negative predictive value. That is excluding the possibility that you progress to Alzheimer's disease. So these are still very preliminary data, but they indicate that besides high-tech and expensive measures, such as many of the biomarkers, these kind of things, which basically cost just the amount of work for doing the testing, have quite good clinical value. Okay, so this was the preliminary data for Alzheimer's disease. Now I would like to point out to the contribution of, I would say, dedicated testing. That is not taking, as we used to do, tests coming from education or developmental psychology and apply them to neurodegenerative conditions. But the idea to develop tests which are targeting a specific issue on the basis of what we know about the normal anatomy of cognition. And I'm going to talk about this group of disorders which involve language and social cognition. Why it's so important to have a specific diagnosis? Because, as I mentioned, this group of phenotypes, that is the language phenotypes and the behavioral phenotype, are frequently due to other disorders, not to Alzheimer's disease, and in particular to this frontotemporal spectrum, which includes a number of different pathologies and is characterized by a sort of probabilistic relationship between the pathology and the phenotype. At this point, usually there is a big discussion starting about what's the use of having a specific diagnosis if you don't have the specific treatment. But the point is that it's a sort of circular reasoning. If you're not if you don't have the specific diagnosis, there is no way you can run good clinical trials with chances to get good results. Because if you are mixing the pathologies and you are using an amyloid drug to treat people who don't have amyloid in the brain, it's very likely that you're going to get negative results, and vice versa. If you have now, there are several drugs in phase 1 and phase 2 targeting tau pathology. If you are giving these drugs to people who don't have a tau pathology, very unlikely that they have positive effect. So it's not just an academic interest, it's also an academic interest because these patients have provided an enormous amount of information in several areas. Think about, for example, about semantic memory. The study of patients with semantic dementia has provided enormous insights into the organization of semantic memory, but has also some, as people like to say nowadays, translational potential. So let's start with the progressive aphasias. There is a set of criteria which have been proposed to diagnose the different varieties of primary progressive aphasia. This was based on a big effort to have a group of people meeting and sharing their insight and expertise in order to develop a core set of criteria. This was a very, very interesting work, very fun to do, but again, it's important to test them on a new perspective series. This was done on retrospective cases. And this is what we have been doing in a collaboration with several people. In the first place, of course, you must make a diagnosis of primary progressive aphasia. And this is sort of simple. The patient must have a language disorder which is progressing, is not affecting for a reasonable period of time, usually 2 years, other cognitive functions. And these were considered to be very rare. Everything is very rare if people don't recognize it. Now people recognize these conditions, and we see a fair amount of cases referred from several institutions. It's if you have the diagnosis in mind, you find the cases. The patient with progressive language impairment come in different varieties, and 3 are the most common: non-fluent, semantic, and logopenic phonological. Why is it relevant to make the differential diagnosis among these 3 conditions? Well, the 3 forms have a different anatomy. As you can see from this slide, this is VBM plus DTI in the 3 groups. And what you can, if you are familiar with the language anatomy, you can possibly predict the kind of picture of clinical picture you are going to observe. More anterior lesions are associated with the non-fluent variety. This condition is associated with an extensive involvement of the temporal lobe, is associated with a semantic disorder. And this form, which is mostly associated with temporoparietal involvement, is associated with a form of aphasia which might resemble the classical conduction aphasia. Why the diagnosis is relevant? Because the anatomy predicts in a probabilistic fashion the pathology. So if you have a non-fluent agrammatic patient, it's very likely that the underlying pathology is tauopathy. While if it's semantic, it's almost always, the majority of cases, this TDP43 pathology. On the other hand, if you have the logopenic phonological presentation, it's most likely Alzheimer's disease. So you see here the whole range of neuropathology is represented in these people with progressive aphasia. And the relationship is probabilistic between the clinical phenotype and the pathology via the location. Is this clear? Okay, so we proposed a sort of minimal battery for the assessment of these patients, which include these 4 areas. Number 1, you need to have a very careful assessment of spontaneous speech, which should be quantitative and qualitative. Then you should have an assessment of single word processing skills, both at the level of naming and at the level of comprehension. 3, we consider crucial sentence repetition. And 4th, comprehension of syntactically complex sentences. This is the minimal battery derived from the criteria. Actually, things didn't really work as we predicted. Well, at least not exactly. Let's very briefly discuss the criteria, and then I'll show you the results of the prospective study. So in the semantic variant, you expect to have mostly naming and single word comprehension impairment. And especially when the disorder progresses, some impairment of nonverbal semantics. While speech is fluent with anomia, semantic paraphasias. So these are the core criteria for semantic variant. And this fits well. This is the previous imaging data I showed were group data based on MR. These are PET data, FDG PET data on the single subjects. PET is very powerful to give you localization in the single subject. You don't need to average a group of subjects as in the previous images. And you see here a patient with a typical picture of semantic dementia, and you see, or semantic variant, and you see the involvement of the temporal lobe, which is much more prominent on the left side. To study this patient, we developed a comprehensive semantic memory test, which includes several tasks, but actually naming and comprehension are enough. These are data from the prospective studies. This is the percentage of patients which are impaired on naming and comprehension. And you see naming is 100% of the patient with semantic dementia, 33% for the other two varieties, and 70% of the patients are impaired on single word comprehension. That is, all patients except those in the very early stages. While comprehension is spared in most of the patients with non-fluent variety and logopenic variety. So the semantic variant is quite easy to separate from the other. You have this quite focal involvement of naming and single word comprehension. Are there specificity issues? Yes, there are some specificity issues in the case of, for example, mutation of this gene, which has been described last year. It's extremely important because it's the most frequent mutation in motor neuron disease, and if we have time, we'll see the connection with that condition too. And these cases can be atypical, but otherwise, it's a quite clear-cut clinical picture, both from the image standpoint and from the neuropsychological standpoint. The big problem is with the other two varieties, and this is creating a lot of confusion, in particular people who are trying to start clinical trials. Because the non-fluent agrammatic variant has this list of features in which the most important is the non-fluent production with articulation impairment and agrammatism. There might be also, in addition, a defective comprehension of syntactic and complex sentences. So most of the diagnosis is based on what? On the assessment of spontaneous speech. And this is something which most people can't really do in a reliable way, unless they have a very specific experience. Speech therapists typically can do it, not necessarily neurologists or psychologists can do it. And this is the paradigmatic case with the typical, you see that, again, this is a single case with positron emission tomography, and you see the area of hypometabolism, which is very similar to what you would expect in a Broca's aphasia, the vascular Broca's aphasia. With the logopenic phonological variant, you run into troubles from the point of view of the distinction with the non-fluent varieties, because the speech is neither fluent nor non-fluent. Some people call it disfluent, meaning that, again, it's based a lot on your expertise and on the evaluation of spontaneous production. Because you typically don't have articulation impairment, but you must be able to score articulation impairment, and you don't have agrammatism. But you have a lot of pauses and interruptions in your production. So it might give you the feeling that the patient is not actually fluent. So again, this relies a lot on clinical experience. The other feature is defective repetition and the disorder of verbal short-term memory. And this is the typical pattern. So here we have a crucial specificity issue, because in one case, if you make a diagnosis of non-fluent aphasia, most likely the pathology is going to be a tauopathy. If you make a diagnosis of logopenic aphasia, in most cases, this is the diagnosis, the underlying pathology is Alzheimer's disease. And already you have also the clinical level a minimum diagnostic management decision to make. That is, if you are seeing a patient with dementia and you suspect Alzheimer's disease, usually you prescribe the drugs which are approved for the treatment of Alzheimer's disease, which is not the case if the patient has not Alzheimer's disease. Of course, this is not a big therapeutic dilemma, but of course this is extremely crucial from the point of view of organizing a clinical trial. Am I including this patient in a trial for a new Alzheimer's disease drug or not? Okay, so what we've been trying to do is to develop a simple procedure to help people to make the diagnosis. And this has been going through several phases. The analysis of connected speech is complicated. Stephen Wilson in San Francisco, together with Marilu Gorno-Tempini, developed a system to analyze the production during the description of this picture, which is taken from the Western aphasia battery. And there is an analytic grid you apply to the production. It's quite nice, but it takes about 4 to 5 hours to score a patient. So this is clearly not practical for everyday clinical use. So, and I'm skipping this. What we have been trying to do is to apply the whole procedure and to try to see if we can extract some simple parameters which allow to make a differential diagnosis between the two. And the interesting result is that articulation is not good. This was done through several raters in different places, rating the same patient. And what happened is that the agreement about articulation was actually very low. So, and as you might remember, this was the main feature distinguishing non-fluent to logopenic from logopenic together with agrammatism. Agrammatism works, on the other hand, works very well. People are good at judging sentences as grammatical or not. And if you plot just two of the parameters which can be extracted from the complex grid, that is syntactic complexity and the disruption of fluency, which are not articulation disorder, but the hesitation, pauses, premature terminations, people are quite good at separating the non-fluent variety from the logopenic variety. The same applies if you plot agrammatism versus length of utterances. The logopenic and the non-fluent separate quite nicely. So basically what we are now testing in the third phase is whether a simple evaluation, not quantitative evaluation of these two parameters, has the same diagnostic power as the complete procedure. Of course, this remains still quite subjective. So another step we are taking is machine learning approaches on the transcription of the extended production. This is work which has been already published by Peter Garrard using a Bayesian algorithm to classify automatically the patients. What we have been doing is to try to improve it. For those of you who are interested in these sort of things, the traditional machine learning approaches to language analysis is the so-called bag of words. You don't consider the structure. You just consider all the words in the transcript. Together with Cristiano Chiesi, who's a computational linguist, we've been trying to develop an improved approach which also puts some structure. Of course, we like the idea that there is some structure beyond the simple number of words. And these are the structure we've been assessing. And as you can see, they are quite remindful of some of the aspects on which the subjective analysis was working, such as the truncated structure. And with this approach, we were able to have a 92% accuracy in classification. Of course, if this is replicated, this is really nice because you just have to collect and transcribe without having to do the very lengthy and painful procedure of scoring the production. There is also another possibility in order to try to make this more objective. And this is the other possibility we've been working on. It's an Italian version of the Northwestern diagram test. You see a picture, you're given a word, and then you have the fragments of the sentence. And the task is to reconstruct the sentence which refers to the picture in the correct way. And these two seem to work, because as you can see, most of the patient with, this is percentage of correct performance, the patients with non-fluent aphasia are much more impaired than the patient with the logopenic variety. So this seems to be another test with a good discriminative value between these two conditions, which are important to distinguish clinically. So far so good. Now I'm going to show you the part which didn't work. Our original idea was that repetition was very good to distinguish non-fluent from logopenic, because logopenic patients typically have a very reduced memory span. So our idea was that repetition was severely impaired in logopenic aphasia because of the defective memory span. Clinically, this does not work very well. Again, if you have a number of different raters and you ask just them to score the performance on repetition, you don't get very good results. Actually, the only significant difference is that the non-fluent patients are actually worse in repeating syllables of words. But as you can see, the sentence repetition doesn't really differentiate the three varieties. The other disappointing point was, again, in a clinical prospective sample, as compared to selected cases, is grammatical comprehension. In this study, we used Gabriele Micheli's and Rita Capasso's test, and did not work to differentiate logopenic from non-fluent. And what is worse, also the semantic patients were quite impaired on the task. So, of the original criteria we proposed, I would say that just two seems to be quite effective in differential diagnosis: naming and single word comprehension, and the analysis of spontaneous speech. This seems to be the most effective to differentiate the three varieties. So I think I almost finished my time, but I can just show you one example of a similar approach to the other interesting phenotype. Possibly the most interesting phenotype from the clinical standpoint, because it's very difficult to diagnose, and quite frequently is seen by psychiatrists rather than neurologists or neuropsychologists. That is the behavioral variant. Why this is the case? Because the behavioral variant, by definition, is characterized by changes in personality and social conduct, which are usually considered more in the domain of psychiatry than in the domain of neurology. Of course, this is changing because, again, the diagnosis of the behavioral variant has some probabilistic relationship with the pathology. In this case, usually, again, TDP-43. You don't have to read this, I just wanted to point that in the consensus criteria, the early signs of the disorder are actually this dysfunction of aspects of what we call now social cognition. That is, for example, diminished response to other people's needs and feelings, diminished social interest, interrelatedness, and personal warmth. This is quite often the first thing that the relatives complain about. And in this case, the effort, rather than finding the tests which could be adapted for this kind of diagnosis, has been to create new tests, because this is a domain which had for many years been relatively neglected in a clinical neuropsychology. And of course, we now know that this has a complex anatomy. If you look, for example, at one of the tasks which have been used, reading faces, deciding on emotion from facial expression, it doesn't work very well. The patients with Alzheimer's disease are also impaired on this task. While we found some tasks which were relatively specific. I don't have the slide about this task, but basically this is a condition in which the patient sees a series of vignettes and has to decide on the basis of the vignette what the person has in mind. It's a theory of mind test versus a condition in which the patient has to decide which kind of feeling the person is experiencing. And as you can see, this task is quite efficient in diagnosing the patient in a very early stage, and it's relatively insensitive to Alzheimer's disease. Oh, I think I skipped this, and I come to the conclusion. I just gave you some example, but I think it's quite important to underline that nowadays there seems to be a lot of interest in what people call deep phenotyping. And when people talk about deep phenotyping, quite often they refer to high-tech, complex, and invasive assessment. A very crucial, important part is actually the basic neuropsychological assessment. If you make a wrong diagnosis on the basis of neuropsychology, most likely you are not going to get good results from the other kind of examination you have. So this is really a crucial step. A consequence of this is that the testing must really aim to be specific more than sensitive. Think about memory tests. We have a lot of very sensitive memory tests, meaning that they are sensitive to aging, they are sensitive to depression, to anxiety, and of course you don't want this. You want to have something which is specific for an ongoing pathological process. And this aim is closely connected to the possibility to have simple and reliable quantitative measures which are suitable not only for the assessment of Alzheimer's disease, but also for the atypical case, which we can mimic as all the other varieties, and for the frontotemporal dementia spectrum. And this is a list of the people who have been working on some of the data I've been showing you.
Thank you very much, Stefano, for this wonderful talk. Any question? Veronica.
Well, it's more like a request for clarification. So in the first part of your talk, you were showing the data about visual short-term memory and the predictability for the development of Alzheimer's disease. And if I understood correctly, you were showing, I didn't understand exactly what the task was, but I suppose it's something like a delay match to sample task. So you were showing the patient or the subject something, and then after a delay you ask them whether the testing was part of the original set or not.
You mean the binding task or the?
Well, I didn't quite understand what you mean exactly with binding and with shape task. So if
Let me check.
So if you ask in one case to remember just shapes, and in another case
in the other you have to remember the combination between shape
so
and
like
color.
bound shape, like
Yes.
shape and color,
Yes.
right? So can you, and I was wondering whether you have if you go to the next slide, I guess, where you were showing that exactly. The shape task is the one that is most predictive, right? Yes. And do you have any explanation for that? Because I would quite naively, I would say that the binding thing would be much more difficult and for some reason would be much more predictable or predictive of.
The binding is most difficult and has the best negative predictive value. If you're doing well on the binding, it's very unlikely that you have Alzheimer's disease. While the shape seems to be more sensitive and slightly less, might be slightly less specific. It's a visual short-term memory task, and it's easier.
And what's perceptual in your perceptual thing?
What's the?
What's the perceptual condition?
The perceptual is the sort of baseline condition in which you have to discriminate the shapes.
So it's not a memory task?
No, it goes below 4. Below 5, which is considered to be a good predictive value. But these are very preliminary data on a subsample. These are only the data for MCI patients.
Okay, and the last very quick question, are you thinking of investigating other dimensions, like other...
Okay, when we started this, I mean, this is a very large study, and when we started, Sergio Della Sala insisted very much in having also the dual task, because he's sort of keen about it. He used it for many years. Probably now I would do something different, because the dual task, I'm not showing the results because they are not very, very, very good. It doesn't have any good sense. It lacks completing specificity. It's relatively highly sensitive, but it's a bit problematic. Now I would include probably the 4 mountain tests or some other more experimental tests. The people are all also getting the traditional test. That is, in Italy, the most used tests are the logical memory and race list, which are also doing well, less well than the 2 experimental measures. But again, this is only a sub-analysis of the subset of the data. I just wanted to show you what you are doing, but I wouldn't consider this as a definite result. Thank you.
Gabriele? The scoring the performance of patients with primary progressive aphasias of various types is basically a nightmare. So, I mean, if the goal is not simply to say that there's something wrong, but if the goal is to say which variant is affecting any individual subject. Now, I'm especially interested in spontaneous speech, of course, and we've developed many, many years ago a very complicated scoring system that probably certainly more complicated than the one devised in 2010, but it would still be unsatisfactory. Now, my question was, at which stage of the disease did you conduct the analysis that Catricala et al. Have put together? In other words, at that stage, when those subjects were tested, was there any other way to independently make behavioral? I mean, I'm not talking about neuroimaging, I'm not talking about neurochemistry, just based on behavior. Was there any other dimension that allowed you to make the diagnosis? And how did those measures correlate with other more basic language disorders in these patients?
Okay, this is a crucial point. All this kind of work risks very much to be circular, because of course the patient had been diagnosed in one of the varieties. The point, of course, is that the initial diagnosis was made on the basis of a standard neuropsychological assessment, and which does not include the evaluation of spontaneous speech. So the evaluation of spontaneous speech was typically been done by the neurologist by ear, by first impression, which is often the case. I mean, if you look, for example, in the most the most extensive progressive aphasia series, the one from Cambridge. Basically, they didn't have a spontaneous speech assessment. And this is probably one of the reasons why the percentage of logopenic patients has been switching so enormously between the different papers. Because probably until 5 years ago, probably all the logopenic patients were called non-fluent patients. Then, and while in the latest series, the diagnosis has been revised, but the crucial problem remains the analysis of spontaneous speech. Our effort is to try to find a way to make this simple and manageable by people who are not spending their whole life in scoring aphasia tests. But as I told you this morning, originally all the scoring had been done by 3 different people. We had to redo this, to centralize it and to redo this completely in order to have more convincing result. Now what we're trying to test is if it's a simple instruction, just listen carefully if there are incomplete sentences and if there are many hesitation, leave alone the articulation, because articulation is tricky, is good enough to allow a differential diagnosis. But basically the short answer is no.
Is there any further question?
Hi, so we're talking about translational aspect of your research. So what does your research translate into for these patients?
The point I was making is that what is happening is that, for example, for many years in all the grants, the grant calls by the European Union, for example, in the area of neurodegeneration, there was no mention about cognitive testing, or it was considered just to be some sort of routine application of standard neuropsychological assessment. Now there is a lot of interest in developing other measures which are specific for the condition. That is, I'm not applying a raised test for memory, but I'm going to try to find a measure which is specific for transentorhinal dysfunction or for dysfunction of the neocortex of the temporal lobe, versus just using a traditional aphasia test taken from vascular aphasia.
So, but as a consequence of this, do you plan a different treatment for the patient? Do you do something different?
The consequence of this is mostly from the point of view of the diagnostic power, in particular from the point of view of clinical trials, so both pharmacological and non-pharmacological therapy. This is the basic translational approach. And it's happening, I mean, with a lot of effort, but of course there is still a lot of discussion about, most people, and I agree, are against mass screening, for example. There has been a lot of discussion recently, because in the UK there has been an enormous grant given to GPs to use a computerized test, which is the paired associate test. It's not the traditional paired associate test. It's an experimental test based on the work of Trevor Robbins' colleague about experimental models. To give it works on iPad, it's a commercial, and to give it to GPs in order to screen all their elderly subjects. And there has been a very strong reaction to this, because, I mean, at this point you really run into the ethical issue of looking for very early form of a disease you are not going to be able to treat. And it's completely different from the condition in which you are selecting, for example, for a clinical trial. But again, this is an example of something which was an application which is based on an idea which comes from basic neuroscience.
Any further question? No? I ask a very naive question, Stefano. I'm completely running out of business, but what is the difficulty, the very nature of the difficulty with the imaging techniques and so on? I mean, with the high-tech technology in predicting this sort of diseases. I mean, it is just a problem of technology, it is not high enough, or it is rather than we still lack a deep understanding of the underlying biology of the disease. What is your opinion?
The imaging techniques are quite powerful in terms of location, as you all know. They are starting to be power to show some possibility towards molecular diagnosis. And this comes from really high-tech approaches. Basically, there is the work on the relationship between specific pattern of alteration of functional connectivity and specific molecular pathologies. This is an area in which we would be very, very keen to be able to work. The group in, for example, the UCL group in London has been doing some very nice work, finding what they call networkpathies, that is specific changes in connectivity related to the molecular pathology. I mean, this is the next step. Because we are very good at localizing damage, but localizing is, the power of localizing is very similar to the power of neuropsychological testing, or traditional neuropsychological testing. Here you have left temporal dysfunction, as we used to say 100 years ago. The tests are compatible with left temporal dysfunction. And the imaging, if properly done, I mean, quantitative imaging, because just looking at the images is enough just in a very advanced stage. You really need quantitation, both with MR and with positron emission tomography. It's quite effective at this stage, but what we really want to do is to be able to go beyond this and towards the specific molecular pathology. It's happening. There are several developments. High field MR is another promising way to look into this. I just read a grant in which they were making astounding claims about the possibility to have histological imaging in the neurodegeneration with 7T.
Thank you. Gabriele?
Could it be that one of the reasons why we're still trying to come to terms with these clinical pictures is that there is a large intra-syndrome variability? Because, I mean, there are still lots of unclear issues. For example, semantic dementia. Everyone agrees that there is a core semantic damage, but on top of that, there is a whole number, I mean, there is a variety of additional deficits that are present in some subjects and not others, which obviously relate to different biological mechanisms. One of the issues is the noun-verb dissociation, another one is the concrete-abstract dissociation. Some patients have a pure semantic damage from the beginning, others from the beginning have also additional disorders. Now, to what extent are we over-relying on some clinical classifications when dealing with these issues? I mean, I know that there's no answer, but I'd like to know what you think.
No, I agree. I would say this is the other side of the coin. Today I've been stressing the sort of translational usefulness, but it goes also in the reverse direction. As I said, the study of patients with so-called semantic variant, I mean, has given such a big impetus to research in semantics in the last few years, uncomparable to anything else. If you do a PubMed search of aphasia, just put the word aphasia. 10 years ago, it was mostly vascular aphasia. Now, if you put aphasia in PubMed, you get just reference about the neurodegenerative form of language disorder. And these are not clinical papers. It's the fact that I think it's what is really fascinating about this is that when you are studying stroke, the consequences of stroke, you are not sampling the whole brain. You are sampling the territory of the middle cerebral artery, while the rest of the brain, it's not typically affected by stroke, so you don't very infrequently see those. For example, the anterior part of the temporal lobe, with the exception of some tumors of some less frequent form of pathology, basically we didn't know anything about it. While the study of this patient has been opening another window, another flashlight on some parts of the brain. And the same applies very much. I didn't have the time to go into this in detail, but in the domain of social cognition, the reason I find it extremely interesting is that it's getting from a very broad and fuzzy field where you had very, very complex concept, empathy, theory of mind, towards a much more analytical effort, try to fractionate the component of social cognition. Again, as in the case of aphasia, psycholinguistics played a major role. In this case, it's social psychology, which is now playing a very important role. We are working on the social cognition battery with John Cassioppo, who's a leading social psychologist, and is very interested in trying to apply sophisticated measures of social cognition to patients. The problem is that most of them don't work because they are too difficult to be applied to patients. So we've been wasting an enormous amount of time on tests which do not work. But it's still very interesting conceptually.
Okay, if there are no other questions, I think we should thank you very much to Stefano.